So I do want to welcome you all from many different parts of both the state of Connecticut and the country. I want to say a special thanks to those of you joining us here in New Haven because it is absolutely gorgeous outside, and you're making quite the sacrifice by being inside with us on the internet some more for another 30 minutes. So thank you for that as well. If I haven't already, my name is Chris Renton. I'm the Associate Director of Communications and Marketing here at the Peabody Museum, Yale Peabody Museum of Natural History. We're going to try our best to do as many of these programs as possible, these remote programs. The best way to keep up with us is to join our email list if you haven't already at our website. You can also follow us on Facebook, on Twitter, and also obviously on Instagram. I do my best to keep up on all those platforms. I hope you'll be able to follow us there. Next week, we hope to have another one of these talks for you. It'll be all about Spinosaurus. So for you dinosaur lovers, please do join us and tell your friends. A couple, just to give you the ground rules of today's talk. It's going to be about 15 to 20 minutes of a presentation with Elissa, and then we'll do a 10 to 15 minute Q&A afterwards. I appreciate your understanding that we won't be able to get to all your questions. We'll get to as many as we can, and thank you for your patience on that front. So the other thing I want to let you know, the Q&A will be done through the Q&A function at the bottom of your screen. You can ask us questions during the presentation. You can wait until afterwards. We'll try to get through those as best we can. Some of you were kind enough to email me some questions in advance, so thank you for those. And we have disengaged the chat function just to keep things a little more straightforward, but please use that Q&A to submit your questions. So without further ado, now that I've gotten all the boring stuff out of the way, I do want to introduce all of you to Elissa Sorojsrisom. Pronouncing that properly was the biggest test for me today. So, I do want to introduce you to Elissa. A quick introduction, and then I'll let her get to it. Elissa attended Brown until 2016 when she graduated with a degree in biology. While at Brown, she worked with a number of researchers as an illustrator, and I believe that inspired her to take part in and complete the natural science illustration course at the Rhode Island School of Design. Next, she worked as a research tech in the lab of Yale Peabody curator Erica Edwards. She began work at the Peabody in earnest in 2018, brought on for work on a grant through the Western Interior Seaway Thematic Collecting Network. Her illustrations have been featured in a number of journals and publications, many of them quite illustrious, including Nature Communications, Royal Society B, Systematic Biology, and the Yale Daily News and Yale Scientific Magazine, just to name a few of them. In the fall, she will begin a joint PhD program at Columbia University and the New York Botanic Garden, studying plant morphology and evolution. I'm pleased and privileged to introduce her and to maximize the remaining time we have with Elissa before she leaves us. So with that, I'm going to leave it to her, and she'll take it away.
All right. Thank you for that lovely introduction, Chris. So when I work with scientists, my job as a scientific illustrator is to help them communicate the outcomes of their research in a way that's easy for their audience to understand. So to show you how I do that, today I'm going to talk about 3 different projects that I've illustrated in collaboration with scientists. And I have a lot of material to cover, so I'm going to get right into it. Hopefully you can all see my screen. Okay, so for this first project, I'm going to focus more on the advantages of using illustration, and then I'll talk more about the process of making the illustrations for the next 2 projects. So this project I did in collaboration with Dr. Jordan Cheney at Brown University, where I also did some of my independent research. And there he was studying the plagioptegyales, which are muscle fibers that are embedded in the wing membranes of bats. So in this illustration here, they're shown by these colored lines. And I know bats are getting kind of a bad reputation in the press right now, but I'd encourage you to keep an open mind about them. They're really incredible animals, and we use them to study all sorts of things from disease to neurology, evolution, and of course, flight. They are the only animals that use a soft wing membrane to fly, and that's why Jorn was interested in studying them. And I always show this video whenever I get the chance because I think everyone should have the opportunity to see a bat flying in slow motion. And the video credit goes to David Burma, who is also part of that lab at Brown. So here is a photo of a bat wing photographed with light coming through it. And you can see some of the structure here, especially in the arm wing, is visible. This is the same wing image under cross-polarized light, which is just a technique that scientists sometimes use to help them visualize biological materials. So these photos were taken by Dr. Cheney while he was collecting data for his research. And our goal with the illustrations was to use these photos that he collected and illustrate the muscle and elastin arrangements in each of the 18 families of bats. And we decided we wanted to include the body of the bat so that the viewers could see the full anatomy of the animal, not just the wing. And I decided to do the illustration in graphite because I wanted to be able to capture a high degree of detail, which graphite is very good for. So this project is older and I don't have a lot of progress shots to show you, so I apologize for that. But here you can see I've illustrated the body of the bat, but I haven't done any of the detail in the wing yet. And I did use that mechanical pencil for the majority of this part of the drawing. And this is a different illustration, but you can see that I've added in the muscles using colored pencil, and I'm just drawing in those elastin fibers using that mechanical pencil again. So the ultimate goal of these illustrations, the reason why we decided to illustrate instead of using a photo is because a photograph like this is very informative for a scientist. They can collect all sorts of data from it. You can see, I forgot to point out earlier, the muscle fibers in the arm wing here are these thicker bands that are lighting up, and then this faint webbing pattern in the wing are the elastin fibers, which is another structure that Jordan was describing. So this is great for the scientists, but if your audience is perhaps not an expert in bat anatomy, they might have trouble interpreting this photo. For example, they might have trouble distinguishing between the different sets of muscles in the arm wing over here, or they might mistaken some of this other anatomy like vasculature for elastin fibers. So by contrast, when you have an illustration like this, even a casual observer can quickly see that there are multiple sets of muscles in the wing, each represented by a different color. And you can also see, easily see the arrangement of elastin fibers in the rest of the wing. And it's also easy to distinguish between the elastin and the muscle because the elastin is in plain graphite and the muscles are shown in color. So this is a Jamaican fruit bat, and the muscle arrangement you see shown here is more or less indicative of what we see in the majority of leaf-nosed bats like this one. And it's completely different from this bat, which is a Mexican or Brazilian free-tailed bat. You can see that the muscles in this bat are very thin and packed very close together. And researchers often describe this as actually a sheet-like muscle anatomy that is pretty unique to this particular bat. And Dr. Cheney was the first person who was describing the muscle and elastin arrangements in many different species of bats. So one of the main roles of these illustrations is to allow researchers to look for connections between the muscle anatomy and the things that we already know about the bats. So this bat in particular Actually, we recently discovered it broke the record for fastest flyer. So while you might be familiar that peregrine falcons can dive at over 200 miles per hour, they can only power their own flight, like flapping flight, at around 60 miles per hour. But these bats have been measured flapping at almost 100 miles per hour. So now that we have this information about this pretty unique muscle anatomy in these bats, it naturally leads scientists to speculate whether that muscle anatomy has anything to do with the bat's ability to fly very quickly. And that's a possible avenue of future study that someone could pursue in the future. This next bat is called the greater bulldog bat, or the fisherman bat. It uses those giant hind legs to scoop up fish. And you might notice that its legs are very beefy compared to the average bat leg, because it needs to be able to generate power in order to lift its prey out of the water. But curiously, it's completely missing this group of muscles that usually comes off of the leg. It was shown it's shown in green in the other illustrations. So it's completely missing those muscles. And the rest of the muscles in its wing are quite thick, but also very sparse compared to what we saw in that Mexican free-tailed bat. I'm going to show just one more example. This is a mouse-tailed bat. These bats usually live in the desert, and they've actually been discovered living in Egyptian pyramids. And this bat has almost the opposite thing going on from that fisherman bat that I just showed. It has lots of these green muscles coming off a teeny tiny leg over here. And that's another thing that a scientist could be interested in looking into, whether there's any sort of correlation between the amount of musculature in a bat leg and the number or amount of muscles that are inserted onto the leg and then go into the arm wing. So hopefully that gave you just a little taste of how illustrations like these can help researchers make comparisons, form hypotheses, and maybe even lead to future studies, while also allowing non-expert audiences to appreciate the nuances of anatomical diversity and also understand the outcomes of these kinds of scientific studies. All right, moving right along to the next to the next project. This is a fossil reconstruction of a horseshoe crab. Lemuridae is the family of horseshoe crabs, in case you're curious. So I did this illustration in collaboration with Dr. Russell Bicknell. He was actually a visiting researcher to the Peabody Invertebrate Paleontology Division, where he was studying some of the fossil horseshoe crabs in our collections, and that's how we met. So he asked me if I could assist with some of the reconstructions of the fossil horseshoe crabs that he was describing. And this is a photo of the fossil that the illustration I showed is based on. Here's a slightly better look at the fossil. It's this orange bit, so you can see the midline here, the body with some spines on the sides, and the head, and the right side of the head is missing. So this fossil is from the late Devonian of Pennsylvania, making it about 380 million years old. And don't know why I can't, okay, sorry about that. And this is a sketch that Dr. Bicknell's colleague in Italy sent me to help with the reconstruction. So the goal of a fossil reconstruction is to depict the animal as it would have looked like in life. And so the first step of the process is to use the photos of the fossil and any materials like this one that the researchers have, and for me to come up with a preliminary sketch that kind of gives the researchers an idea of what I'm thinking. So I don't know why. All right. This was my first draft. So I sent this to the researchers and they sent this back to me. This is actually a really common way that I receive feedback from my collaborators. They'll often just print out a sketch that I've sent them, draw on top of it, and then snap a photo and email it back to me. So here they're trying to show me that I need to fuse these two lobes up here, get rid of this line and add a hinge to the tail, and also probably shorten the tail and make it a little thicker. So with that in mind, I came up with a second sketch. And this one was a little closer, but there's still some things that we needed to do. So Dr. Bicknell sent me this figure that he was preparing for the paper. And here he's showing me there are some new anatomical features that they want me to add in, like this notch in the back of the head and also this anatomy at the base of the tail. And we also need to make sure that we get the number of lobes correct. So with that in mind, I come up with a third draft of the sketch. I added in all those anatomical features, and the researchers were happy with this sketch. So once I have a preliminary sketch like this, the next step for me is to come up with a shaded version of that sketch or a value study. And I do that just so that I can use it as reference while I'm making the final version of the illustration. And I want to show you this photo series that I took. This was actually for a different horseshoe crab reconstruction project, but I continue to use it for all of my illustrations of horseshoe crabs because it's really great lighting reference. So this is a specimen from the Peabody's invertebrate zoology collection, and I photographed it with light coming from the left side of the specimen. In scientific illustration, it's best to have light coming from one side or the other so that you have a clear light side and a clear dark side, because this helps describe the topography of the surface better than if you had just direct overhead lighting. And I've also imaged it at 3 different exposures. You might notice the one on the left is very dark and the one on the right is by anyone's measure overexposed. But the reason I do this is because cameras capture a narrower range of light than our eyes can see. So in order to get the full range of detail and shadow information that I need to come up with a realistic looking drawing, I need to take multiple photos at different lighting settings. So using these photos for reference, I took that sketch that I had and I just made a shaded version of it. This one I just happened to make using a digital painting program in my iPad. And this is just a look at the evolution of the sketch from the first draft on the left through the second and third drafts, and then that shaded value study on the right, which shows me the dimension of the specimen. So for the final version of this illustration, I used a technique called stippling, which is essentially using many tiny dots to shade a drawing. And it's just about as thrilling as it sounds. So I'm gonna speed up this video so I don't bore you to death. Stippling is a really great technique for black and white illustration because it creates a very smooth looking gradient from light areas to dark areas. So essentially what happens is if you view a stippled image from far away, it creates an optical illusion where your eyes kind of just blend everything together and it looks very smooth. And this is an advantage over using lines to shade, because lines can sometimes make people think that there is some sort of surface texture or maybe markings that are not supposed to be there. So stippling is a very nice technique for avoiding that. And here's just another look at the final version of the illustration. If you're curious, they were publishing on this specimen because they were reassigning it from the genus Bellinuris to Piquetia based on some of those new anatomical features that they were describing. So for this last project, unlike the other 2 projects, it's also a fossil reconstruction. But unlike the other 2 projects I've talked about, instead of using traditional media like pen and pencil on paper, I created this illustration completely digitally using my iPad. So this is Sorojsrisom Cthulhu. It might remind you of a starfish, but it's actually an ancient sea cucumber ancestor. And this fossil is from the Herefordshire Lagerstätte in England, and it's about 430 million years old. So I created this illustration in collaboration with the curator of invertebrate paleontology at the Peabody, Professor Derek Briggs, and his colleague at Oxford. And this is the sketch that they sent me to help with the reconstruction. So I believe one of them just made it using his computer. And they also gave me a few CT scan printouts. So Professor Briggs always prints out the reference that he gives me, which is really great because he hand annotates notes onto them for me, and that is just fantastic. You can never give an illustrator too much information. Usually we don't get enough information. So here he's showing me that this fossil has different levels, if you will, of legs, and he's labeled them A, B, C, and D. So this upper level here are the A level legs, and then going down. And then here again in the CT scan, he's trying to help me distinguish out those C level legs. So for this project, I actually have the full process videoed, and I'm just going to talk over it while it plays. So the first step, like in the last reconstruction, is for me to come up with a preliminary sketch that gives the scientists an idea of what I'm thinking. And my goal is to include as much detail as possible so that they understand what I'm going for, using as little drawing as possible so that if changes need to be made going forward, I haven't wasted all my time adding in details that are going to be removed later. So this is the first sketch that I sent to the researchers, and Professor Briggs came back and told me that the scales on the top level, that A level of legs, is actually different than the scales on the rest of the legs. So I made that change, and then we also discussed how the scales are not flush with one another, they're actually overlapping. So I've also gone in and redrawn the scales on one leg from each level to show that overlapping anatomy. And this is the second version of the sketch that I sent in. And the feedback on this round was that now we really need those lower legs to look more three-dimensional and look like they're holding up the body off of the ground. So here I'm just adjusting, I'm adding that leg in, and then I'm adjusting the lower legs so that they look like they're weight bearing and that they're making contact with the surface of the ground and lifting the body up. And I also omitted this anatomical feature, the periproct. So I'm just using the CT scan image as my reference, and I'm adding that back in. And then at this point, I decided I was really unhappy with that upper A level of legs. I wanted them to look more three-dimensional and dynamic. So I'm just redoing them one more time. And then I'm finally ready to add in all the scales. So normally, I might not take the time to make a very clean sketch like what I'm doing here, but when I have a subject that's this complicated, it's really great when I go in with color to be able to work over something very clean. So that's my finished line art, and I sent it to the researchers, they okayed it, and I'm finally ready to add color. So you might remember that this fossil was named Solacina Cthulhu after the Lovecraftian god, and the researchers specifically requested that I use green and purple and that I make it look somewhat ominous and menacing. And this fossil is actually, it's a little secret. It's only a few centimeters across, but I think they really wanted me to make it look grand in order to be worthy of the name Cthulhu. So here I'm just using green and purple, trying to make it look like the kind of starfish colors that we're used to seeing in extant starfish. And then I'm just adding in the background using a picture of the seafloor as reference. And here's my final version. And just to show you how much of a collaboration these illustrations really are, this was the version of the painting that I sent to the researchers. And Professor Briggs' colleague wanted it to look even more dark and menacing, so he actually edited the file himself to make it darker, and that darker version was the version that ultimately got published. So it was a true collaboration from start to finish. So that's all I have for you. Thank you so much for tuning in. I'm going to turn it back over to Chris, and we have time for some questions now.
Fantastic. Thank you very much, Elissa. We have a number of questions, so I won't waste any time, but I think I'll try to categorize them into three groups. The first one is sort of specimen-based, and we'll answer those real quick. Do you happen to know, this question is from Mia, what is the horn-like shape on the head of the Jamaican fruit bat? Do you happen to know?
Oh, was it the nose? So I believe you're talking about the nose leaf. So that Jamaican fruit bat is part of a family of bats called Phylostomidae, and most of them have that nose leaf. It's actually it's evolved in those bats to help them with echolocation.
Oh, fantastic.
And that's about all I know about them.
Okay. Mia, if that did not answer your question thoroughly enough, you can always contact me, and we'll get you some more information. The other sort of specimen-related question comes from Hans. He asked if the fossils you're working with are compressed during the fossilization process. How do you know how to reinflate them when you're reconstructing them artistically?
Mm-hmm. So we use a number of different techniques for trying to figure out what a fossil would have looked like. One is just to look at the way it's been smushed, because sometimes you have some clues as to what the shape would have been based on the pieces and how they broke. We also use either other fossils or other examples of animals that we know are kind of either closely related or closely resemble the fossil that we're trying to describe, and we use those to help inform how it might have looked if it wasn't crushed. But there's a certain amount of guesswork that goes into it.
Okay, that's fair. Thank you. So I kind of want to move into how we're getting a number of really great questions that all center around, well, there's a bunch that center around your process, which I want to get to in a moment, because I think that'll be really helpful for the folks who are listening who want to break into this field or who want your advice on how to improve their work. But there's a question that came in from a Mary. She said, I have college degrees in both art and biology, but have no idea how to get started in scientific illustration. She's wondering, how does one break into the field and find commissions? And real quick, to add to that maybe would be like another question that came in from Susie, which is, how do you market yourself? So how do you break in and how do you stay in?
Okay, so this is a big subject. I would say that most scientific illustrators do struggle to get started. It's kind of a difficult field to break into. Usually what happens is you just start out by doing just small projects for people that you happen to know or that people put you in touch with. So when I first started, I just was lucky. I was at a university, so a couple of people approached me for jobs, but I really only did one or two a year. And I still really only do one or two a year. It's not my full-time job. I know that's really not inspiring at all, but the truth of the matter is it's actually a really difficult field to break into, and it's even more difficult to make into a full-time career. The best way to market yourself really is to find a way to get your work in front of researchers, if that's who you want to work with. Otherwise, scientific communicators, so that's a broader group of people that you can target, but there's no website or any sort of platform that's the best place to post your work. The best thing to do is somehow show your portfolio to the people that would actually be the ones hiring you. And I don't have specific, well, I don't want to I don't,
Go ahead.
I don't really have like specific advice for how to do that, but I will say that I run a website called sciencefindsart.com that is specifically for this purpose, for putting your work in front of scientists. So you can
I think you're allowed at least one personal plug, giving a webinar at 4:30 on a day. So thank you for that comprehensive, your attempt at like a comprehensive answer. You're very honest, and I hope the folks who asked those sorts of questions, if you have any follow-up questions, please direct them to me.
Yeah, I will say you can also, Chris, you can direct them to me and I can give a more full
thank you
answer over email. Yeah.
That's very kind. So, you know, so on the reverse of that, how, and maybe you sort of covered this, but I think one of the other questions we're getting is like, in your experience, how did scientists find you? Like if a scientist is looking for a great illustrator, where do they go to look other than your website?
If only they went to my website.
Sure.
So this is a funny thing about this field. I think scientists actually have trouble knowing where to look for illustrators. So the way that I've gotten work, by far the most common way for me to get work is through word of mouth. So again, while I was at Brown, someone would mention that I did scientific illustration, and then I would have researchers email me out of the blue asking, because they actually don't know where to find illustrators. So when they found out that there was one nearby, they got really excited. And the same has been true at Yale. Like I mentioned, Dr. Bicknell was visiting Yale, the paleontology division, just for his research. He was just there to look at our fossil collection, and happened to find out that I did scientific illustration. And he actually wanted to hire me without even seeing my portfolio, which is, I shouldn't say, but it's true. Like he is just like, I really need an illustrator. This would be great. Will you please do this? So yeah, it's really getting to know the people.
Well, and I was, on that note, I was going to suggest to the folks who are with us who asked those questions, or who are interested in finding, you know, getting into that community. I am the furthest thing from a paleo artist, but because I run the Twitter account for the Peabody, I've noticed that there is quite a community of paleo artists online, and I'm sure once you've established a specialty in that community, like you said, you know, people will start recommending you for particular projects, I would imagine, right?
Yeah, recommendations are really big, because scientists will ask each other if they know an illustrator. So getting your work out there, that's a good point. If you can put your work on social media, that is one way to kind of get noticed, but it's not a surefire way to kind of kickstart a career.
Sure. So on the closer to your, I think a lot of people have questions about your process. So to get that started, we've got a few questions coming in about length of time for these pieces. You know, how much time are you devoting to them? And then also, what is that time being spent on in terms of software? Like, what are you using for digital digital software? I'm forgetting the terms they use, but you know what I mean.
Yeah. So in terms of how long it takes me to do a piece, I don't tend to time myself, but I do know that last project that I showed, the fossil sea cucumber that looked like a starfish, that took me 60 hours because the program timed me, so I know. And I would say the majority of that time, you might have noticed this was true for both processes that I showed, a lot of the time is spent on the sketching phase, because in scientific illustration, it's important that everything is correct. So you spend a lot of time revising and lots of time going back and forth. And then once you finally get that underlying sketch, you kind of just do the rest. And that actually takes, I would say it tends to take me less time once I get to that stage. Uh, and the bat drawings, I would say they maybe take me around, I want to say 20 hours to finish.
Okay.
And the stippled horseshoe crab, probably around the same, maybe a little less. But stippling is very time consuming, so maybe not. And you asked about the software I use. So I have an iPad Pro with an Apple Pencil. So that's what I use to draw when I'm doing digital work. And I use the program called Procreate, which I think remains to be one of the most professional programs that you can get in terms of just what you're able to do on the iPad. Although now you can screen share with your computer onto the iPad. Like you can use it as a second desktop basically. So now you can actually have Photoshop, which can also be used for digital painting.
There was a quick, can you repeat your website? Someone asked.
Oh yeah, it's called sciencefindsart.com.
Perfect. Thank you. There were like 3 or 4 people who asked that. And thanks everybody.
I'm embarrassed. It's down right now because I'm updating it. So I'm sorry. You can't actually look at it, and I'm really sorry.
A couple of folks are now saying they went to their website and it looks like it's under construction. So.
It's been up and we recently took it down. I'll put it back up in the next few days. It'll be back up.
No worries. No one seems upset. There were a couple questions. I think people always are interested, especially if they respond to an artist's work or an artist's practice, about inspiration. Are there particular paleo artists or maybe particular pieces that drew you into this world?
Oh, wow. So even though I showed a lot of paleo work, I actually, most of my work is not paleo. So one of my earliest and probably continued inspirational artists is Justine Lee Hirten, Hirten. I can't remember her handle. You should be able to find her. She is a contemporary scientific illustrator. Of course, like all the classics, Audubon. Albrecht Dürer was actually a quite prolific natural science illustrator, and his work is pretty interesting because often he would illustrate without seeing the animal. He would just come up with an illustration. So that's interesting.
And fairly easy to find on the internet too.
Yeah, exactly. And another like favorite would be Harris Ray Ching. He's more of a wildlife artist, but it's another favorite.
Can you repeat that real quick? I think we lost you for like a second.
Harris Ray Ching.
Okay. Fantastic. Thank you for that. You're getting, by the way, I just want to, there are a number of folks who have foregone asking a question simply to compliment your work. So you're getting a lot of lovely compliments in the Q&A.
I appreciate it.
I think we've got time for about one more question. So I'm just trying to find one really quickly. And yeah, 1 second. There's quite a few coming in. So here's a good one from Zoe. She was wondering, do you find that you create pictures more digitally or more on paper? Do you lean one way or the other? And then how do you? Another person had asked, like, how do you determine your approach when you enter a project?
Mm-hmm. So I am partial to traditional media, basically using pencil, pen, or watercolor on paper. That's my favorite way to work, but the field is very rapidly moving into preferring mostly digital work. So I would say when I do my own work, it's usually traditional, and when I do work for clients, it's mostly digital. And the way that we determine that has a lot to do with the turnaround time, which I will say is generally not enough time. So digital tends to be faster just because you can make changes much easier than you can with traditional techniques.
Mm-hmm.
And it's also a lot of the work that I end up doing, it either needs to be reproduced or it needs to look kind of diagrammatic. So in that case, I would use vector illustration and Adobe Illustrator to do the work. So if they need anything sort of diagrammatic or anything that needs a lot of tiny adjustments so we can get it right, it's better to do it digitally because it'll just get done faster. Yeah.
Great. And you're at liberty to not answer this question, but we have had a couple emailed, and then a couple have shown up in our Q&A chat. When you work with these scientists, are you getting commissioned? Are they paying you a commission, or are you?
Mm-hmm.
How does that process work for the folks who want to break in?
Mm-hmm. So I am, other than some of my early projects that I did as a student that ended up counting for like credit for my classes, I have always been paid for my work. And I would encourage anyone who's going to do scientific illustration to demand that they are paid for their work, because it's a problem in the field that some people will do work for no pay. And you really shouldn't do that. Your work is worth something, and you should always ask to be paid for it. And also, I would encourage, on the flip side, if you're a scientist and you want to work with an illustrator, you can get funding through the NSF or through grants. You can just write in some money for scientific illustration. It's part of the way that you're going to communicate your work, and it's a great thing to put in your broader impacts. So,
Fantastic.
Write illustration into your grants.
That is a great. I think that's a great way to wrap this up. It'll help everybody find work. Thank you very much, Elissa, for joining us, and for your presentation, and for all the knowledge and information you shared with us. We've enjoyed having you. I've enjoyed working with you, and I know you'll go on to do even bigger and better things in the fall. The last thing I'll say to the folks watching, if we didn't get to your question and you'd like it answered, please feel free to write directly to me. My email address should be in one of the emails you received when you registered. We'll try to track down an answer for you. I'm sorry we didn't get to it. But I do want to say again, we are going to continue doing these online programs. Please follow us on social media, sign up for our email list, and reach out directly with any questions, concerns, recommendations, requests for programs. We're all ears, and just want to say thank you to you, Elissa, and to everyone watching. We'll see everybody soon.
Bye.
Bye.